This is the first lecture in the skeletal muscle series, Structure to EC Coupling. I am Vincent Barnett. The objectives for this series will cover muscle organization and structure, the nerve-muscle communication, excitation, contraction, coupling, what's going on inside muscle cells on the molecular level as muscles contract, some mechanical aspects, and muscle metabolism. This was the schedule for spring. Let's start here. Voluntary movement is mediated by the shortening of skeletal muscles, so that the way the muscles attach to the skeleton will allow the movements of the limbs. Muscles are often organized in what we call opposing pairs. In an opposing pair situation, such as the biceps and the triceps in the arm and forearm, when the biceps contracts on the front side of the arm, on the anterior side of the arm, then what you see is muscle flexion, that is, the forearm moves up towards the arm. Flexion moves the bones of the forearm closer to the bones of the arm. The triceps muscle, which is on the back side or the posterior side of the arm, has a tendon that wraps around the elbow and attaches to the ulna of the forearm, so that when this muscle shortens, it pulls on this bone from the posterior side and then extends the arm, pulling these bones of the forearm away from the bones of the arm. The triceps muscle shortens and the limb extends. Note that when the biceps contracts, the triceps is relaxed, and when the triceps contracts, the biceps is relaxed for these movements. Occasionally, you would activate both muscles at the same time, and through that activation, rather than flexion or extension, what you would get is maintenance of the arm's position. So you, for example, you're carrying a bag of groceries into the house, the bag's heavy, you're holding it from the bottom. You keep both arms locked by activity of both the triceps and the biceps. These are some terms I would hope you will learn, and we will use them throughout this series. That a muscle cell can also be called a muscle fiber. A cell membrane in muscle sometimes is called a sarcolemma instead of a plasmalemma, because sarco, a Latin term that means flesh, is often used to refer to muscle. By that reasoning, the cytoplasm can be called the sarcoplasm, and the endoplasmic reticulum can be called the sarcoplasmic reticulum. And in skeletal muscle, the sarcoplasmic reticulum plays a special role in the activation and relaxation of skeletal muscle, which we will discuss later. Now let's look at this organization of skeletal muscle. If you pick a muscle group in the body and examine it closely, what you would see is that that muscle group is composed of fascicles, and a fascicle is a collection of individual muscle cells. The fascicle is surrounded by a sheath of connective tissue, and that sheath runs the full length of the muscle and connects to the tendon at each end, so that it helps to communicate the forces that are generated when the muscle contracts. If we look between the fascicles, you can see that that's where blood vessels and nerves will be running. And if you look within the fascicle, what you will see is that there are individual muscle cells or muscle fibers within the fascicle. You find that each of the individual muscle cells is also surrounded by a sheath of connective tissue to separate it from the other muscle cells, and that the muscle cells themselves in skeletal muscle have a very distinct appearance for a couple of reasons. One is that they have multiple nuclei. There can be hundreds or thousands of nuclei in a skeletal muscle cell, because as muscles are formed, as the embryo is turning into a fetus and as the body is maturing, the muscle cells form from precursor cells that are called myoblasts, and these precursor cells fuse together to form a larger cell. Because these cells become so large, you end up with hundreds and thousands of fusions, and in each case the nuclei are retained, so the final mature cell has hundreds or thousands of nuclei. And they're distributed along the length of the muscle cell. This provides muscle a unique advantage in that because the tissue is so active and because of the activity there is the potential for muscle cell damage, any damage can be repaired locally using information from a local nucleus. And so this distribution of nuclei along the muscle fiber allows muscle to recruit the nucleus that's nearest the injury, to use the genetic information there to replicate the proteins that are needed to rebuild the muscle cell, and to have that repair accomplished more efficiently. So you have this hierarchical structure. That just means there's multiple levels. From a muscle fiber, an individual muscle cell, these muscle fibers are bundled into fascicles, which are groups of muscle cells, and then the fascicles are bundled into a muscle group in the body, which we often name based on either its location or its function or its appearance. So whole muscle has this multiple layer structure. Under the microscope, you also notice that skeletal muscles are striated, that is, they have stripes that are perpendicular to their length. These stripes are due to structures within the muscle cell. On the left we show a cartoon version, on the right a micrograph, a picture of a segment of muscle cells, and you see these striations that go across the long axis of the muscle cell. You can also see several nuclei in this image and the connective tissue that runs between the cells. So there's something inside these muscle cells that leads to a striped appearance, and this turns out to be important for muscle function. So let's take a look at what we see when we go inside. This diagram shows the interior structure of a muscle cell, and one of the things that we see in the interior of the muscle cell is that it is packed with these protein-based assemblies that run the full length of the muscle cell, and these smaller fibers, fibrils, are called myofibrils, the myofibrils within the muscle fibers. The myofibrils are striated, they have stripes, and the stripes are due to the density of proteins that are found in different regions of the myofibril. You have dense regions dominated by structures called thick filaments, and less dense regions which are dominated by structures called thin filaments, and they repeat along the length of a myofibril, and this is what gives muscle its striated appearance. Another important feature is that the myofibrils, the individual myofibrils, are aligned so that their dense regions are in parallel with the dense regions of the adjacent myofibrils, so that they're all in register across the width of a muscle cell. We also see in this picture mitochondria, nuclei, and we notice that the muscle cell membrane has little holes in it, and these holes are connected to a network that runs through the center of the muscle cell. This is a membrane-based network. You can think of it as just an extension of the plasma membrane or sarcolemma, and it's called the transverse tubule network because these tubules run across the muscle cells, and they surround the myofibrils, as you can see. Adjacent to the transverse tubular network, you see another membrane system, and this is the sarcoplasmic reticulum. The sarcoplasmic reticulum is also in intimate contact with the myofibrils. All of these structures play a role in the activation and relaxation of muscle cells, which we will explain in a few minutes. For the moment, let's focus on the myofibril. Within the myofibril, you see that there is an area that is designated as the sarcomere. Here's that word sarco again. Sarco meaning flesh, mere meaning body. So sarcomere is a muscle body. It's a structure within the muscle cell. The sarcomere has two distinct regions, the A band, dominated by the thick filaments, and the I band, which is dominated by the thin filaments. You will also notice that the A band is bisected by a line, an M line, and the M line joins the thick filaments together. It's actually a three-dimensional disk or network that links these filaments together. And also at the in the middle of the I band you find a Z disk or Z line, and this connects the thin filaments. In the center of the A band there is a zone that's slightly less dense, and it's called the H zone. It is the interaction of these filament systems that then helps to control muscle length and force generation, and we'll develop that story as we move through these slides. Now, let's find out a little bit more about the proteins that make up these structures. So if we start with the myofibril, we know that it is made up of sarcomeres that are stacked end to end running along the length of the myofibril. Within the sarcomere, which we define as the space between two Z disks, we find the Z disc at one end, actin thin filaments that project towards the center of the sarcomere, and then we have the Z disc at the other end and actin thin filaments projecting in the opposite direction towards the center of the sarcomere. In the center we find the thick filaments, and they are joined together at the M line. These thick filaments are tethered to the Z lines by a filamentous protein called titin. Titin runs as a single polypeptide from the Z line all the way to the center of the sarcomere from each side, and it is the largest protein in the human body. These thick filaments are made up of a molecule called myosin. Myosin actually has 6 subunits, 2 very large ones that are sometimes called the myosin heavy chains, and they have globular head domains and long alpha helical coiled tails. The coiled coil tails of 2 myosin molecules will wrap around each other, and then that assembly will form the backbone of the thick filament. There are 4 small proteins called myosin light chains, and there are 2 per myosin globular domain, and you can see them here just at the base of the globular domain, the 2 myosin light chains. These proteins, the light chains, have a modulatory role in skeletal muscle contraction, and they are absolutely required for the control of smooth muscle contraction. An interesting aspect of the thick filament is that the myosin molecules are arranged in an anti-parallel arrangement, and that is, these globular domains or heads are projecting in opposite directions on either side of the M line, so that the myosin thick filament can reach out and interact with actin on either side of the sarcomere, and then interact in a way that helps to generate force that will be balanced across this distribution. The thin filaments are primarily made up of a small protein, relatively small protein, called actin. Relatively compared to myosin, the molecular mass of this entire myosin assembly is about 500,000 units, and in the actin it's about 40,000. So, individual actin molecules will assemble into a filament. This filament, it looks almost like a twisted strand of pearls. It's also alpha helical in its twist. And on the actin thin filaments you also find some accessory proteins. One is called tropomyosin. Tropomyosin, shown here, is this coiled coil assembly. It's a dimer of 2 filaments and runs over the length of 7 actins. And then for each tropomyosin there is a complex called troponin, and troponin is 3 proteins, troponin T, troponin C, and troponin I, and they help to confer regulation to the actin thin filament. We'll get to that in a bit. Tucked into the middle here you see a protein called nebulin, and nebulin, like titin, is a single polypeptide and runs the full length of the actin thin filament. So these filamentous assemblies are spontaneously formed in the cytoplasm of muscle cells. They create sarcomeres. The sarcomeres then are built so that they go from one end to the other, and at the end of the sarcomere, there are connections that run through the plasma membrane out to the tendon to communicate forces to each end. Now, that's a lot of information about how skeletal muscles are put together in terms of the structure of the cells and the structures of the sarcomeres and the myofibrils. And one way to keep it straight is to build yourself a flowchart. And this is the flowchart recommended by the textbook, which starts with skeletal muscle and shows that it's composed of fascicles, and that in each fascicle you have individual muscle fibers or cells. And then there are components within each of these cells that include myofibrils, sarcoplasmic reticulum. Depending on the cell, you may also have mitochondria or glycogen storage, and that these myofibrils themselves are made up of a collection of proteins, the thick filaments primarily made of myosin, the thin filaments primarily made of actin, and then there are these associated proteins of titin and nebulin. This is one way to look at it. I tend to organize my thoughts in a slightly different way, where I go from myofibrils, which are made of sarcomeres, and then the sarcomeres composed of thin and thick filaments, and then the filaments composed of the proteins. And so you can choose how you want to organize this information for your own use. However, let's go back to this, the one that the book uses. There's no right or wrong here. It's just how is it easy for you to keep all of these components straight. So this is what the book chose. This is how I modify it. Excuse me. This is what the book chose. This is how I modified it for the way I think about the structure of muscle cells. This is a more general structure, but you can make your own diagram, whatever helps you keep these components straight. Here's a summary of the properties of a myofibril. Myofibril is a contractile element in a striated muscle cell. Forces are generated within the myofibrils, and those forces are then communicated to the tendons and help muscle contract. They are organized in parallel in muscle cells. They are composed of sarcomeres. They terminate in a myotendinous junction. This is the connection that goes out to the tendon through the muscle cell membrane. They fill almost all of the cytoplasmic space in a striated muscle, which means that some of the organelles are pushed out to the periphery, like the nuclei, and you can see some mitochondria pushed out to the periphery. That's because almost all of the space is taken up by the myofibrils. Something that we hadn't talked about yet, but it's very important: muscle strength is proportional to the number of myofibrils in a muscle cell. That is, as cells get stronger, they gain more myofibrils. You synthesize more proteins, you build more myofibrils. Your muscle cells have to accommodate them by getting bigger, and bigger muscles, bigger in diameter because they have more myofibrils, are stronger. If you go on a period of bed rest and your muscles start to atrophy or weaken, you lose myofibrils. So muscle strength is proportional to the number of myofibrils. And that these myofibrils are surrounded by the transverse tubular network and the sarcoplasmic reticulum, those membrane structures. This is an electron micrograph of a sarcomere showing the A bands and the I bands, and a cartoon derived from this structure. So it was electron microscopy that helped us define the structure of the sarcomere. Now this is a longitudinal section. Longitudinal section along the length. What happens if we slice this and look at it end on? Well, the first thing to note is it depends on where you slice it. Depending on where you cut through a muscle cell or a myofibril, what you will find is you will sample different parts of the system. If you are in the I band, you would get actin only or primarily. If you're in the H zone, in the middle, you would get myosin thick filaments only. If you're right at the M line, then you would see that connection between the thick filaments within the M line. And if you're in that overlap zone between the actin and myosin thin and thick filaments, what you find is how the actin filaments and the myosin filaments are arranged relative to one another. And this turns out to be very interesting and very key to the structural basis of muscle contraction. So let's look at that overlap zone. And what you find is that there's some geometry going on here. The structure is almost like a crystal. To make it a little easier, I've drawn a diagram. And what you find are, for each actin filament, there are 3 myosins that can interact with it. For each myosin filament, here's a myosin filament, there are 6 actin filaments surrounding it, and that means that each myosin can project a cross bridge out to any one of these 6 actin thin filaments. And then also, the myosin thick filaments are arranged in a hexagonal order. This order allows some physical measurements to provide information on the structure of muscle cells. You can determine the distances by looking at diffracted X-rays that will bounce off of these structures. We call it a double hexagonal array. And it's amazing that a biological tissue builds this very crystalline structure, and that structure is functional for muscle contraction. Muscle contraction actually turns out then to be pretty straightforward. During muscle contraction, the thin filaments will be pulled by the myosin cross-bridge domains into the lattice of the myosin cross-bridges, so they move towards the center of the sarcomere. This movement shortens the sarcomere, and since the sarcomeres are stacked end to end, the whole myofibril shortens, and because the myofibrils are connected to the myotendinous junction and to the tendons that connect to the bones, the muscle shortens and the limbs move. When this occurs, you would note, if you were looking at micrographs of these muscles, that the dimensions of the A bands stay the same while the dimensions of the I bands would contract. As the thin filaments slide into the sarcomere center, past the thick filaments, the I band appears to shorten, or does shorten, and it's only because these filaments interdigitate or move past one another. Now let's see if we can move on and describe a little bit about why this occurs. Go back to our membrane systems. We have the sarcolemma or plasmalemma, that's the muscle cell membrane, the transverse tubules, which move through the center of the muscle cell, and then the sarcoplastic reticulum. These are all integral elements. When muscles are activated, they generate action potentials. These action potentials occur along the membrane, as you learned when you discussed the action potentials in the nervous system. When they reach the transverse tubule network, they move into that network and move through the center of the muscle cell. This is important because as muscles get stronger, they get thicker. And if the activation signal was only on the periphery, then you would see that the outside rim of the muscle cell would start to contract while the inside still hadn't received the signal. Because the transverse tubules move through the center of a muscle cell, that means the signal doesn't just ride on the surface of the cell, the signal runs through the muscle cell and it can activate the entire cell essentially simultaneously. Now, just because an electrical signal is moving along the membranes of the transverse tubule, that doesn't activate muscle contraction by itself. It activates muscle contraction because it runs past the sarcoplasmic reticulum, which is a storehouse for calcium, and it makes the sarcoplasmic reticulum releases calcium into the cytoplasm, and in the calcium itself, that is the signal to trigger muscle contraction. So that's a reason for the sarcoplasmic reticulum to be in such close contact with the myofibrils where the force is generated. There's a coupling between the electrical signal in the transverse tubule and the calcium release from the sarcoplasmic reticulum that we'll discuss in just a few slides. So this shows a network of the transverse tubular system and the sarcoplasmic reticulum, the lateral sacs full of calcium, and also it's important to remember that there will be a need to remove that calcium from the cytoplasm when the muscle relaxes, and so there are also pump proteins within the sarcoplasmic reticulum that can reclaim the calcium after a muscle contraction. Let's look at the activation of the skeletal system by the nervous system. Motor neurons in the spinal cord project axons out to the skeletal muscles. And as they approach the muscles, the axons divide and then innervate a collection of muscle cells. A motor unit is defined as a motor neuron and all of those muscle fibers that it innervates. A single muscle cell will only be innervated by one motor neuron. And as the diagram shows, in our bodies, you can have cells that are adjacent to one another that are innervated by different motor neurons, making them parts of different motor units. This provides a level of control for the activation of muscles and the generation of force. All of the muscle fibers in a motor unit will be of the same fiber type, and so many of you have heard of fast and slow muscle. In a fast muscle motor unit, all of the cells are fast muscles. In a slow muscle motor unit, all of the muscles are slow muscle cells. I mentioned in class that there have been some interesting experiments that show that the communication between the motor neuron and the muscle cell help to tell the muscle cell which genes to express, and that if a slow muscle cell is has its neuron removed and replaced by one from a fast muscle cell, it transforms some of its proteins to more mimic a fast muscle phenotype. Here's a reminder from your studies of the nervous system that the nerves that control skeletal muscles are called the somatic motor system. They have their cell bodies in the spinal cord and the central nervous system. They project axons out into the body that will synapse on muscle cells, communicating with those muscle cells through the use of acetylcholine. Acetylcholine will activate nicotinic receptors, which will then activate the skeletal muscles. You've seen acetylcholine used before in the parasympathetic system. It is the communication chemical at the synapses of the ganglion as well as at the target organs, which can be smooth and cardiac muscle and some glands. In the sympathetic system, it is only at the ganglion. It is also the molecule of communication within the adrenal medulla of the adrenal gland. This is an image of the neuromuscular junction of skeletal muscle shown in a scanning electron micrograph. So you can see the surfaces of the neuron, the footprint of the axon terminal as it sits on the surface of the muscle cell, and you can see that it's highly specialized at that interaction point. This cross-section shows transmission electron microscopy showing a nerve terminal full of acetylcholine vesicles, the Schwann cell that's enveloping and insulating the nerve terminal, and the muscle cell which is underneath and has a specialized contact area that's enriched in those nicotinic acetylcholine receptors. This is a cartoon diagram. You have the active zones where the vesicles fuse. The postsynaptic membrane has a lot of invaginations or infoldings which increase the surface area so to ensure that you get reception of the signal. And muscle contraction is initiated by a nerve action potential which causes vesicle release at its nerve terminal, opening, diffusing across the cleft, binding to the nicotinic acetylcholine receptor, and opening the ion channel of the nicotinic acetylcholine receptor at the motor end plate. That's what's called the postsynaptic side of this synapse is called the motor end plate. And the opening of this nicotinic receptor causes an end plate potential. Not an action potential, but an end plate potential. The nicotinic acetylcholine receptor is nonspecific. It does not only conduct sodium, does not only conduct potassium or calcium, but it can conduct all of them. It's a cation channel, and that just means that its opening allows the movement of positive ions down their concentration gradients. However, the net influx of sodium helps to depolarize this area and cause a graded potential, which then starts to spread from the center of the neuromuscular junction and out towards the surrounding area of the membrane, where it encounters voltage-gated sodium channels, and the opening of the voltage-gated sodium channels is what triggers a muscle cell action potential. That action potential moves along the membrane and propagates through the T-tubular network. At the bottom of this diagram, I've drawn a cartoon of how this might occur, where the blue dot is the neuromuscular junction. Once the nerve releases its acetylcholine at the neuromuscular junction, you get a local depolarization which spreads. It's a graded current. When it encounters the area where you find the voltage-gated sodium channels, that's when you get an action potential that spreads in both directions. Oh, here's another way of looking at it. You get a depolarization at the neuromuscular junction followed by a spread of an action potential in both directions. What's the consequence of this action potential spread? Well, first, here's your nerve action potential, very rapid depolarization in the nerve cell membrane. It triggers a muscle cell action potential, which is a little bit broader, has a slightly different shape due to the different characteristics of the muscle channels, voltage-gated channels. And that results in a contraction phase in the muscle followed by a relaxation phase. You'll also note that the depolarization occurs before the contraction and relaxation. The electrical event occurs before the force-generating event. That means there's something else going on in the muscle cell in response to the electrical signal that then triggers the muscle contraction. The end plate potential spreads passively until it encounters voltage-gated sodium channels. This opening of the voltage-gated sodium channels causes the muscle action potential. The muscle then has a single, for a single action potential, will have a single contraction relaxation cycle, and this latent period is this short delay between the action potential and the beginning of tension development. And here's where the sarcoplasmic reticulum's calcium comes in, because calcium is released from the sarcoplasmic reticulum and it binds to troponin on the thin filament, and it's that process that actually allows myosin and actin to interact in a way that generates force. So let's go back to our diagram and note here in our transverse tubule we have this dihydropyridine receptor, DHP, which has a plus on it, which means that it is, in this case, a voltage sensor, and it will sense the depolarization. We get an action potential. It spreads along the muscle membrane. When it encounters the DHP receptor is in a class of proteins that we would consider an L-type calcium channel. But in skeletal muscle, it's not really acting as a calcium channel. Mainly it's using its voltage sensing ability because it is coupled, if you note here, it's closed, a closed RYR, that's the ryanodine receptor, that's the calcium release channel. When the action potential passes the dihydropyridine receptor, it causes an opening of the ryanodine receptor release channel, and the calcium can diffuse out. There's no electrical signal passing between the transverse tubule and the sarcoplasmic reticulum. It's purely the physical interaction, and so I consider this mechanosensitive opening of the sarcoplasmic reticulum's release channel. Calcium is released because calcium is in such a high concentration inside the sarcoplasmic reticulum and such a low concentration in the cytoplasm of the relaxed muscle cell. Calcium flows down its concentration gradient and rapidly rises in concentration, saturating the troponin sites on the thin filament and facilitating muscle contraction. How does it do that? Remember, this is the structure of the thin filament. Troponin C has a calcium binding site. Troponin C is very much like calmodulin, which is a soluble calcium binding molecule that can activate a number of enzymatic pathways. In this case, when troponin C binds calcium, it changes its shape, and that shape change is communicated to the tropomyosin, and the tropomyosin is sitting on the myosin cross-bridge binding sites on actin. So myosin cannot interact with actin in a force-producing way in relaxed muscle because tropomyosin is blocking its interaction site. When the calcium binds to troponin C, it facilitates the movement of tropomyosin away from that site, and then force can be generated. So, calcium is released, it binds to troponin C, the tropomyosin moves out of the way, and then myosin, which was blocked in relaxed muscle, is now able to bind to actin after calcium has bound to troponin. And then myosin, which has previously bound an ATP molecule and hydrolyzed it to ADP and phosphate, can use the energy that it has trapped from that hydrolysis to power a conformational change that occurs as phosphate is released from the binding site on myosin, the active site on myosin, causing the actin filament to move. This process can occur in a cyclical fashion as long as the calcium concentration is high. So ATP will bind to myosin, relaxing or releasing myosin from actin. Myosin rapidly hydrolyzes ATP, but unlike most enzymes that catalyze a chemical reaction, myosin holds on to the ADP and phosphate in a way that traps energy in the myosin head. In this state, it is not rigidly bound to the thin filament. Despite what this cartoon says, it actually can't interact with actin very strongly in this state, with ADP and phosphate bound, in part because of the tropomyosin that's blocking the force-generating site that it needs to bind to. In the presence of calcium, myosin can bind to that site, it releases phosphate, it releases the energy that it's trapped because of the hydrolysis, and it can then move the actin filament. Phosphate release initiates the cross-bridge power stroke, and ADP release is essentially the end of the power stroke. After ADP is released, myosin and actin are still tightly bound to one another. We call this the Riger state, or the state of rigor mortis. Excuse me, we call this the Riger state. We do not call it the state of rigor mortis. Rigor mortis occurs only when the tissue dies. Because in living cells, ATP is always present, and ATP will allow the cycle to repeat. ATP binding to myosin will reduce its affinity for actin, It can then hydrolyze that ATP to ADP and phosphate. If there's calcium in the system, it can go through a power stroke, and then it can repeat this cycle over and over again as long as the calcium concentration is high. If the calcium concentration starts to decrease, then tropomyosin will slide back into a blocking position. So myosin is completing a power stroke, moving the actin-thin filament. The relaxation of skeletal muscle occurs when calcium is pumped back into the sarcoplasmic reticulum by a calcium ATPase, a calcium pump protein that uses the energy of ATP hydrolysis to pump calcium against its concentration gradient into the lumen of the sarcoplasmic reticulum. Once it's inside the lumen of the sarcoplasmic reticulum, that calcium is rapidly bound by a protein called calsequestrin, taking it out of solution, which helps to assist pumping of calcium out of the cytoplasm into the sarcoplasmic reticulum. Without the calsequestrin molecule, the calcium would be too high of a concentration and would actually crystallize within the sarcoplasmic reticulum. This reduction of the calcium in the cytoplasm will then cause the release of calcium from troponin, and then tropomyosin will move back into a blocking position, and myosin will be in its ATP or ADP phosphate states, which are where you would find myosin in relaxed muscle. One of the consequences of this contraction, because of the independent action of the myosin cross bridges, is you always have a probability that you could get more cross bridges formed in one half of the sarcomere than the other, and if that happens, you can get sarcomere disorder where the whole A band shifts towards the Z line in individual sarcomeres in a very asymmetric way, and that's not a way for muscle to propagate good contractions at the beginning of each contraction signal from a neuron, and so you need something to reestablish the order. The way the order is reestablished is that each of these thick filaments are tethered to the Z line by titin molecules, which act as little springs on the end of the thick filaments. So if a band shifts towards let's say it shifts towards this Z line or Z disc on the right side of this sarcomere, the titin is stretched on the left side of the sarcomere, and when the muscle relaxes, that potential energy of the stretched titin can be used to restore the A band to the center of the sarcomere, so you always start this tug of war between the left and right side of the sarcomere with the myosin in the A bands centered in the center of the sarcomere. So we activate by releasing calcium, we relax by removing calcium. We find that the process of generating force and the process of relaxing muscle both require ATP, so those are two roles of ATP in muscle contraction. The third role of or use of ATP in muscle contraction is that disassociation of myosin from actin at the end of the power stroke. If you don't have ATP, you're locked in that position, and that is what we would call rigor mortis. This slide summarizes the sequence, and you should be able to walk yourself through the sequence, how you initiate a muscle action potential, how that action potential then causes calcium release, and how the calcium release triggers the interaction between actin and myosin, and then of course what happens to relax the skeletal muscle. Let's end here. This is a video that's available through the website of our publisher, and I hope that this has been a good way for you to learn about the structure of skeletal muscles.